So next is about motility determination. Before going into it, what is motility? Or motility means what? It is the ability of an organism to move from one place to another by means of flagella, cilia, or special fibrins like which produce gliding motion. So the bacteria which shows movement or motion, they are called motile bacteria, and they use flagella, which is a thread-like locomotive appendage or a structure extending outward from the plasma membrane and the cell wall. There may be either single flagellum or the multiflagella. And the bacteria that does not show any movement or motion, they are termed as non-motile bacteria. However, sometimes in aqueous solutions or in water, this non-motile bacteria shows some erratical movement. This erratical movement is due to the Brownian movement present in the present in it. This Brownian movement is a result of a random movement of water molecules. So during this random movement of water molecules, will also bombard the bacteria, which will cause them to move. So we will think of it, they are in the they are showing motion or they are moving. This will give an erect result or false result for us. So this browning, so sometimes this non-motile bacteria also shows motion, that is due to the Brownian movement. Then coming to the motility determination, it is a method used to identify or differentiate the motile or non-motile bacteria, and it includes three methods, and it is based on the pathogenic capability of the organism. And they are, so it is based on, for the non-pathogenic organism it includes wet mount slide and hanging drop slide, and for the pathogenic organism it includes soft agar stabbing, or also called tube method. First is wet mount slide. First method is and this method is for non-pathogens. This is the simplest way to determine the motility of the bacteria, and it is done by placing a loop full of organism on the slide and cover it by the cover glass or cover slip. Well, in addition to determine the motility or the presence or absence of the motility of the organism, it is also help us to determine the cellular shape and the arrangement of the bacteria, that is irregular cluster pairs or long chains like that. We can also uh, it also help us to identify or determine the arrangement or the shape of the bacteria. Then wet mount preparation is done by, first is by taking a clear or clean glass slide, then place a drop of water. If we are taking a bacterial sample from the agar, then place a drop of water on the middle of the slide, and before that label the slide with the name of the organism. Then place a one drop of water in the middle of the cover glass slide. Then take a few full of bacteria and spread it in the water. Then cover it with using a cover slip carefully without tapping any air bubbles. Then after that, first observe under the low power, then followed by high power. So this will help us to see the motile or non-motile bacteria, or what's to say, or the cellular arrangement and shape of the bacteria. Then after this, we have to take care of some important steps, that are, here we are using unstained bacteria. So we already know that these bacteria are colorless and highly transparent, so it is difficult to see. Therefore, while focusing, reduce the lighting sufficiently to make the organism visible. So reduce the light uh, according to the organism is visible to our eyes. Then next thing is, for the true motility, look for the directional movement, that is several times the long dimension of the bacterium. The movement will also occur in different direction in the same field. Next step is, ignore the Brownian movement, which will make erect or false result in our observation. And next thing is, always examine the wet mount immediately once it has been prepared, uh, because the motility decrease with the time after the preparation. So wet mount preparation should be examined immediately after the preparation, or it will decrease the motility of that organism. Next, the advantages of this method is, quickest means for the determining motility, then useful for determining the cellular shape and arrangement. These two are the advantages of. Then disadvantages are, the slide quickly dries out and organism becomes immotile. So this is the simplest method. So the slide gets dried up very fast. So it is one disadvantage. And if the organism is pathogenic, there is the possibility of the danger to the person in handling the viable organism on the slide. And wet mounts can't be stored over a long period of time, and this slide method can't be stored for long period of time, since uh, the slide gets quickly dries out. So these are the advantages and disadvantages. Next is, second method is hanging drop method or hanging drop slide. So the organism is observed in a drop that is suspended under a cover glass in a concave depression slide. It is like this. So this is a depression glass. 1 minute. This is the depression glass slide, and our organism is and our specimen in the drop, which is suspended from the cover slip, is this. So this glass slide have a concave depression at the at the middle, and the slide for the hanging drop is grounded with a concave well in the center, and the cover glass holds a drop of the suspension, which I showed before. When the glass cover is inverted over the well of the side, a drop hangs from the glass in a hollow concavity of the slide. So it forms. So water drop. This drop is in a glass chamber. Since the drop lies within an enclosed glass chamber, drying out occurs very slowly. So, since it is in a since forms a glass chamber, so the drying up of the slide will be slow down. Help us to slow down. A ring of Vaseline around the edge of the cover slip keeps the slide from drying out. So, a ring of Vaseline is also applied around the cover slip. This will prevent the what drying out of the slide. Then hanging drop slides are useful in observing general shape of living bacteria and the arrangement of bacterial cell when they are associated together. Now, method or the steps involved in it are: take the uh, glass slide with a concave center, and take the specimen uh, from the culture of 15 to 20 milliliters, and place it in the center of the cover slip. Then before that, uh, using petroleum jelly or Vaseline, uh, cover spread the Vaseline or petroleum jelly on the four corners of the cover slip using a toothpick. Then invert uh, ah, then invert this uh, um, invert this glass slide over the cover slip, so the concave well is facing downwards, and fix generally press it for fixing the cover slip over the glass slide. Then slowly turn back, so the uh, concave wall is now facing upward, and place it over the microscope and observe under the microscope in lower power followed by high power. This is the hanging rock method. And here also we have to note down some important step, that is, always examine hanging drop preparation immediately, because the organism become less motile with the time. Then particular care must be taken to avoid breaking the cover slip, since it is more vulnerable when supported only around its edges. And always make sure the specimen is on the top side of the slide. These all are the some important uh, things to note down during the hanging drop experiment. Now, now before so this wet mount slide and hanging drop experiment both are for non-pathogenic organism. Now next is method for the pathogenic organism, that is soft agar stabbing or tube method, otherwise called as tube method. Since the pathogenic, we know that these pathogenic organisms or bacteria are very dangerous, so it is dangerous to attempt to determine the motility of this pathogenic organism by slide technique. So it is very dangerous to determine their motility by using wet mount slide or hanging rock experiment. So that's why we are using this soft agar stabbing. So in this method we will use a SIM medium, which stands for sulfide indole motility medium. SIM medium. For this, for soft agar stabbing technique or method, for this method we will use a special medium called SIM medium. So which is a combination of differential medium that tests three differential parameters, that are sulfur reduction, indole production, and motility. So these three parameters we can determine from one single medium. That is the use of this medium. Then this medium has very soft consistency that allows the motile bacteria to migrate readily through the causing cloudiness. So if the bacteria is a motile, then this bacteria forms cloudy in the medium. that represents or that shows the motility of that bacteria. And the non-motile bacteria will only grow on the soft agar tube only area where they are inoculated. If the bacteria is non-motile, then they will grow only the area where they are inoculated. But in the case of non-motile bacteria, they will grow along the area of stab and will also sim out away from the stabbed area. The negative result is detected by the growth in distinct stone directly along the stab. And a positive result is indicated by the diffused or cloudy growth mostly at the top and the bottom of the stem. So this is the method. This is the soft agar stabbing or tube method. Now steps involved in it are: first we have to label the tubes of SIM medium or semi-solid medium with the name of the organism. Then inoculate the needle and the neck of the tube. Then remove the cap of from where the medium or sample is taken. And then sterilize it and stab it 2 by 3 of the way down of the bottom, and stab this 2 by 3 of the way down of the bottom. Then flame the neck of the tube again and returning the cap to the tube. Then incubate this tube for 24 to 80 48 hours 24 to 48 hours at room temperature. After that, examine the SIM culture for the presence or the absence of the black precipitate along the line of stab inoculation. A black precipitate of FES indicates the presence of H2S. That is, we already said that this SIM is a differential medium, so this medium can be used to identify sulfur production in sulfur reduction, indole production, motility. So if H2S is present, then the black precipitate will be formed, or absence or presence of black precipitate will will indicate the presence of H2. And if desired, one can also test for the indole production by adding 5 drops of quartz reagent to the SIM culture and looking for the development of red color at the top. So we already said that this medium is a differential medium which is made up of sulfide indole motility medium. So we in this single medium we can use for determining the uh, sulfur reduction, indole production, motility determination. So if the sulfur reduction is taken place, then the black color precipitate will be present, that is due to FES. And if it is checking for the indole production, it is done by simply adding 5 drops of coax reagent. So if the red color is developed, it indicates the presence of indole production. And for the motility determination, the medium will become cloudy or diffused. So observation here, this is the positive. I will show. This is the positive test. This becomes cloudy and red. This shows this shows indole production, also the motility of the bacteria, motile bacteria. So result in agar stab, well depressed growth from the line of inoculation, which is evident for the motility, and the presence of the black precipitate indicates the H2S is produced, that is for the sulfide reduction test and the presence of red coloration in the medium after the addition of Kovacs reagent indicates a positive indole test. So one single medium can be used for three different purposes. That's all. Thank you.